Okay, online people, can you hear me? Yes. Okay, so we'll better get started. I think so. The plan today is we are going to finish applied biology and all the past paper questions that has been asked from it. So there's a chance in your year there could be up to 2 MCQs and maybe one third will or a bit less, right? A bit less than one third of a structured essay could be asked. There could be a short note as well. Yeah. So up to maybe 5 to 6 marks could be from applied biology. Okay. So that's why we are in this last moment. I decided to cover that topic as well. So today we'll be finishing applied biology. Okay, so from next week onwards, we are going to go with a question-based revision from past papers. Okay, at the revision class. So I'm going to be teaching some theory topics together with because in the revision class we didn't cover excretion. Okay, we didn't cover excretion. So I'm going to finish quickly that theory next Wednesday as well. Excretion quickly I'll cover urine formation, excretory systems, and sort of regulation, osmoregulation, those kind of things I'll go through again. Because in endocrine system also we talked about osmoregulation, but we didn't cover excretion. So I'll do that next week as well. Okay. So then this Friday we are continuing our past paper discussion. I'll be discussing the remaining MCQs. Okay. And also we'll be discussing some select questions from the SCQ as well, some difficult parts of the SCQ as well. Then next week it's going to be 2022 paper. Okay, 2022 paper we'll discuss. I think we'll have time to discuss 23 and let's try to discuss 24 as well. Okay, so that's the paper class program. It's going to be past papers mainly. So it is up to you to complete the past papers and join for the discussion and assess if your marks are slowly increasing, because these are the final, you should be doing your final studying at this point. Okay, so study the entire week and before Friday's discussion do the past paper and check whether if your marks are increasing. Right? Study the entire week and do the past paper and check whether your marks are increasing. Okay, every week you should increase your marks. Yes, that's what we are doing. So 21, 22, 23, 24, up till 24 you should increase your marks, right? Whether it's, I mean, your goal could be different at the level you are, your goal could be different, right? It could be you can get for MCQs above 45 at home, right? But in the exam your MCQ would be maximum above 40, because in that situation and the paper could be difficult. So practice at home to get above 45, you are going to get 40 at the exam. If you practice to get above 40 at the at home, you are going to get 35 at the exam. That is a reality of the exam. So at home, practice to get above 45. That is the, then in the exam you are going to get the so it's going to set off your chance of getting an A. Okay, because above 40 is like our benchmark for an A, no? Okay, so for a B you need 35, up beyond 35. For a C in the MCQ, I'm saying, because the essay is the place where everyone is going to go from an A to a B, B to a C, because the structured essay could be difficult, the marking scheme could be difficult, but MCQ scoring should, it's the best place to score. Yeah, if you can get above, I mean, get a good score in MCQ, your mind would be, I mean, you don't have to stress much about the essays, because the essays could be something you haven't done in a long time. If you get like a hidden topic, if you get as an essay, you can't help if you haven't studied that. But the MCQ paper is from the entire syllabus now. Yeah. So if you, even if you go to the exam with a just a general idea, you can mark the MCQ. That's what I'm saying. Be prepared for MCQs and see how your MCQ marks are improving at these days. You have almost exactly 1 month left. Exactly 1 month left. Yeah, a lot of things can be done. Anyway, we'll have a small discussion as we go along. Okay, we'll go to the final part of your food preservation and food sort of biology related to the food processing. Okay, so we'll come to the topic called post harvest losses. Okay, there's a definition, please highlight this. the losses that occur along the food supply from harvesting until its consumption. Okay, so until you eat something from the point of view cutting something, imagine you harvest an apple from a tree. From the moment it is plucked from the tree and until you eat it, it is considered as, I mean, during any of that time period, you can get a post harvest loss. Okay, from harvesting to consumption, there is a nice gap. So, from harvesting, you have harvesting methods, then you have transport, then you have temporary storage, then you have again transport at a certain store in the fridges or something, then you are exporting or something in a ship, and then you bring it home, and in your home, you store it. And in your home, you do some preparation, you cook it or something, right? Not apples, but maybe vegetables or whatever. Then until you eat it, food post harvest losses can occur. So remember this definition, from harvesting to consumption. Okay, then let's keep reading. Product quality at harvest is influenced when and how the harvest is done. There are pre-harvest factors. There are pre-harvest factors that it's going to determine how the crop is going to come out as, such as the seed and the quality of the seed. Okay, because what you harvest is what you're going to reap. Right? What you harvest is what you're going to get. So if you didn't harvest, I mean, if you don't, what did I say? If you plant something good, you're going to harvest something good. Okay, you're going to harvest what you, no, you're going to reap what you sow. Right? So you have to plant a good seed or a plant if you want to get a good harvest. So that's why they say source of the seed and its quality has to be good if you want to get the good quality harvest. Okay. Then the quality of the harvest could be lost. What are the places? During harvesting, handling, transport, storage, and domestic processing and distribution. So the order is a bit weird. So what's going to happen is, yes, you harvest, then you handle, yes, then sometimes you store, then you transport, then again you sometimes store, okay, then you distribute, okay, then it comes home, then again you sometimes store, yes, then you consume. It can be complicated, right? The journey of a food item. Okay, so so many places, right, during harvesting. So we are going to see what are the several situations during handling, right? All the situations where losses can occur. So just have a general idea. Okay, have a general idea. They could be asked in MCQ. This is just general knowledge. Okay, so post harvest losses could be a quality loss, a nutritional loss, a seed viability loss. That means if the harvest you are using it as seed again in the next season, you are going to plant your harvest as seed. The seeds might not be good quality. Then commercial loss, you are going to lose money. Okay, so all these, because the quality loss due to weight loss, nutritional loss, because of all that, you are going to get a commercial loss, no? Because you can't get money for the crop as much as you expected because the quality is not there or it is spoiled. Okay. Okay, so let's see what are the post-harvest losses that are possible in cereals, mainly in Sri Lanka, paddy, fruits and vegetables. We are not going to consider about meat items and those kind of things. Okay, we are going to consider cereals, fruits and vegetables. Okay, right, let's see. What can happen during harvesting? You can harvest them at the wrong time, right? It is not the right time to harvest. So let's see. Paddy should be harvested at the best time considering the variety, right? So you shouldn't do either. You can't wait until it's too late, or you can't harvest it too early. So you have to harvest at the correct time. So untimely can mean before the proper time, or you can say too late, too early or too late. Okay, so those are the two ways: too early or too late. Okay. What are the things you can have? Quality loss, nutritional loss. Again, the things we talked about: weight loss and commercial loss. Okay, highlight. Okay, right. Those losses could be further increased. Paddy fields are flooded due to heavy rains. Those are natural hazards. Okay. Then, if harvested earlier, can you just keep this in mind? These are general knowledge. In the MCQ, they can ask what would happen if paddy is harvested earlier. It's going to require more drying. Right? There are tetavi kela you might have heard. Right? These large scale mills, they don't usually buy tetavi. You have to dry them. Have you seen when you're traveling to Anuradhapura, those kind of or narrow areas, they are drying paddy on the road? Yeah, that is because they have to pay these large scale mills to dry them. So they are deducted from your money the farmer is going to get. So that's why they dry in the sun. Then high moisture, what can happen? Mole growth, fungal growth, and insects, right? Who can eat the grains? Okay. And low milling yield means you are going to take the seed out of the husk, no? Potteng eliyata gannawa. So that is going to be reduced. Okay. Depending on the method of harvest, part of the paddy crop may be left and plowed into a soil, which is a commercial loss. So this is something that is happened due to the machine you used to harvest. Okay, so there are things called harvesters. Have you heard of these combine harvesters or anything? So different harvesters harvest in a different way. So if they don't cut it properly in the machine, some of the crop may be left in the field. Okay, so you are losing some of your harvest. Okay, then what about fruits and vegetables? They also should be harvested at the best time. Okay, to have the maximum quality and also to obtain the highest quantity. Okay, if what's going to happen if you harvest too early? Okay, the crop will not be matured enough, right? Ripe or matured enough, then you can't consume it. Got it? Then if it's harvested too late, it may spoil too. These are general things, no? Kaling gathot idila mati, right? It's not harvested, sorry, ripe enough. If you harvest it too late, it might go bad too soon. So you can't store it or transport it for a longer time. So it could lead to quality loss, nutritional loss, economical loss. Okay. Uh, it should be done without allowing physical and mechanical damages to occur. Obviously, you should be careful when you're harvesting. You shouldn't drop all the apples on the ground. Have you seen how they harvest apples? Right? They harvest apples in a commercial level. So if this is the apple tree, okay, they around this sort of the stem of the apple tree, they are going to sort of hold something like an umbrella that is going to sort of go around it, and using a machine, they are going to shake the apple tree. Okay, so all the apples are just going to fall softly on this sort of it's like a rubber cloth, or like a tarp or something, like a tent material. So when they are shaking the tree, all the apples are going to nicely drop onto this rubber sheet. Okay, so that's not going to damage anything. What would happen if you damage the fruit skin or something? Right, storage life will be reduced, spoilage with microorganisms, because they can quickly invade. Keep this in mind. What would happen if damages occur? Insects, microorganisms can invade through the damaged area. So what we just talked about was what's going to happen if you don't harvest properly. Okay, what happens if you don't harvest properly? Okay, then during handling, okay, so as soon as you harvest something, then you have to handle it, you have to package it, okay, you have to make it ready for transport. Those things should be done. So when you are doing, what can happen doing those things, what can happen? Okay, let's see. High shattering loss of paddy seeds could occur, which is a uh, commercial loss. Shattering loss means when you are handling, the paddy seed can break down. So inside the husk, the paddy seed has become powder because you are dropping it, you are crushing it, the lorries are going over it, people are walking over the harvest. Because of those things, the inside the paddy seed of the husk, the paddy sort of what we eat, the starchy part of the seed is crushed. So you can't, it becomes powder as soon as you mill it. Milling means what? You take the seed out of its husk, right? Commercial loss. Harvested paddy should be dried, bagged, and stored temporarily without allowing the paddy seed to absorb moisture and greater attacks by insects and rodents. Yes. So if you usually if they absorb moisture, what's going to happen? They are going to germinate. Okay, the paddy is going to germinate. Yeah. Then you can't eat it. You can't take it to the mill because it's going to send roots like this, right? And they are all going to be connected by small roots. You can't separate the seeds. Okay, so those things can happen. Then, if you turn to fruits and vegetables, they should be handled hygienically. Okay. Of course, you could. I mean, particularly these soft-skinned fruits and vegetables, these things that don't have a very tough skin, right? And a coconut again, you can handle them roughly, right? Something that has a very thin skin, you have to handle them very carefully. Okay, in plastic cartons, which should be kept in the shade temporarily. Okay, should be sorted to remove. Yes, you have to sort them. So if you want to remove the damage or infested moldy, overripe. If you keep overripe fruits together with normal ones, others are going to get ripened as well, right? Because of what? Because of what? What would happen? So imagine you have some raw apples, and you have one ripe apple in between all those apples. What would happen? Yes. Yeah, all the others are going to get ripe. Why is that? I mean, the ripening of all the others are going to be accelerated. You know why? Why is that? Anyone? I'll give a hint. It is due to a plant growth regulator. Yeah. What's the name of that plant growth regulator? Food ripening? Yes. Ethylene. Ethylene is sort of like it's a volatile thing. It's released as sort of like particles, like gaseous. Okay. So ethylene released from this ripe one, yeah, can accelerate the ripening of all the other nearby. So it's kind of used commercially as well for ripening purposes. It's not. I mean, it's not harmful because it's a natural plant growth regulator. Okay. So ethylene can be used in not in Sri Lanka, right? Ethylene, sort of commercial ethylene can be sprayed to accelerate ripening. Okay, so these things just keep them in mind. You have to remove damage, infested, overripe ones, right? You can answer an MCQ from these, right? Right. One of the biggest places where damages occur is due to transportation because you are physically moving it, right, over roads and everything. Poor road infrastructure, improper and poorly maintained roads, right? Raj spillage. Mulimma, the first thing is you are going to spill along the road. Paddy. Okay, spillage. Keep that in mind, the word spillage. And also high contamination. They can get mud in them, rain, right? So many other things that are in the lorry can get into contact. Yes. Multiple movements of the crop. Highlight that. What do you mean by multiple movements of crop? So from one lorry to the other, you move again and again. Okay. So you throw the bags from one lorry to the other or something. That is going to be a big loss. Ametin, bag paddy is loaded and unloaded from vehicles several times before it is milled from one lorry to the other. Some grains are lost at spillage. Okay. Ametin, so 2 to 10% of losses occur in Southeast Asia. Imagine you are milling, if you got a delivery of 1,000 tons, you have lost how many? You have lost 100 tons, up to 100 tons just by spilling. Yes. Okay. So imagine the percentage of loss, such a wastage. Yes. Could be minimized by what? You need to remember then. So these are the problems. Well planned, better modes of transportation with an efficient bulk handling system rather than using people to load and unload. If you can have a sort of machine based loading and unloading, it can prevent these damages. Okay. Okay. During the transportation of fruits, vegetables, arrange at the bottom Okay, so we are moving on to the transport of fruits and vegetables. So you can understand we are mainly focusing on mainly first paddy in all the things, and then we are moving on to vegetables. Okay, so at the bottom, damaged by the weight of the above. Okay, so use of crates that could be stacked. This is something very important. I want you to remember. What is the best solution that you have for the transport of vegetables? If they ask fruits, use of plastic crates that you can stack on top of each other instead of using just these goni, right? Sacks of vegetables will matter that you are going to put weight on. You can use plastic crates, okay, because they are going to take the weight, no, so they are going to nicely stack on top of each other like our containers. Yes, so they don't give weight to the vegetables inside. Got it? So would solve this issue. Use of straw or something soft. That's something you can do, right? So to absorb the sort of the friction and other things, you can have some material in between. Have you seen apples are transported in this sort of sponge-like covers? Yes, apples are put inside this like a net-like mesh of sort of foam like cover, no? So that prevents their sort of bumping into each other and rubbing. Okay, rough handling should be avoided. Uncontrolled changes of temperature, very important in vegetable and fruit transport, and also excessive shaking. It's going to sort of crush the fruit or the vegetable. Okay, what one of the solutions that we do in happens in Sri Lanka, transportation carried out at night time. Okay, if you can transport fruits and vegetables at night time, that happens in Sri Lanka. That's mainly because in Sri Lanka to avoid the traffic. Okay, but it can be protected by the high temperature. Keep that in mind. What is something very, it doesn't even require anything additional to do a transport at night. Okay, simple thing, just transport it at night. Then storage. Okay, another place where due to the lack of knowledge a lot of loss occurs. Okay, so then what about okay, losses at storage losses are considered most critical. Okay, yeah, this is a very important point. What is the place where maximum losses occur? You need to remember, can be asked as MCQ. Maximum losses occur during storage. Can you highlight this? Maximum losses occur during storage. Maximum losses occur during storage. Okay, that's an MCQ point. After, so we are coming back to the paddy story. After harvesting milling, they are stored for short or long periods as food reserves, and for seeds as for seeds for the next season. Particularly paddy, you have seen these large, very tall, what we call as what do you call them? Those large towers of silos. Have you heard silos? Yes, they are stored grain silos in this Prima factory in Colombo as well as in Trincomalee. They have like several 100 feet tall silos for wheat. Okay, then in sort of large scale milling, paddy milling, they have also several massive silos. Okay, so what would happen is rice would absorb the excess moisture, and they can also get infested by insects, right? Certain insects, then that would be a trigger for the spoilage by microorganisms. Then rodents, rats, and other pests. Okay, those kind of things, other damages that can occur during storage. Just keep a few things in mind. Okay, what would happen if you absorb excess moisture? It can get infested by pests, then spoilage by microorganisms, attacked by rodents. Okay, just keep a few things in mind to write in the structure as a paper. Proper storage infrastructure is a solution. Understood? You need to have properly scientifically designed this sort of silos and storage spaces for the long term storage. Then, coming to vegetables and fruits, uncontrolled changes of temperature. Okay, so mostly what happens is due to power cuts, refrigerators break down, therefore food get spoiled. So you need to have backup power. Some you should have a proper plan, right? They should be stored in properly chilled rooms to reduce the rate of spoilage. Okay. Finally, we come to the final stage that so you buy the food from the market or whatever, then you bring it home. At home, what could happen? So at home you can have problems during home storage in your fridge or whatever, and it could be when you are preparing the food, right? So preparing the food uh, can cause spoilage as well. Okay, listen. Uh, okay, so we talked about this, ne? Right. So during domestic processing could be reduced by what? Correct procedures, hygienic handling at home. Okay, hygienic handling to prevent microbial contamination and proper storage. Right. So if you have sort of rice at your home, you should usually store them a bit above the ground so that you don't get the moisture or it could be attacked by rats. Right, in airtight containers. Okay, so that you don't attract moisture. Okay, those kind of things. Then, what can you do at home for the preservation? Few things are given. You can dry once more, right? You can dry the paddy you bought once more, uh, the rice you bought once more at home if you want to keep them for a long time, right? Then thermal treatment of jackfruit where available cost again, right? That means you are going to dry thermal treatment. Okay, you can dry jackfruit, other things. Uh, you can't do pasteurization at home, but we are talking about sort of when you buy them. So you in your home freezer also you can store fish and meat. No, okay. Chilling means at what temperature do you chill? Okay, 4 degrees Celsius. Ne? In the sort of the lower compartment of the fridge. Salting to make lunu dahi, right? Those kind of things. Salting and drying of fish don't happen at home. Homemade jams can happen, right? Chutneys and other things you can do. Use of chemicals, pickles. That is, I think, using what is the chemical used in pickles? Vinegar, eh? Yeah, vinegar, acetic acid is added for the pickling process. So just have a general idea. This is all general knowledge. Okay, any questions? Let's see if we can have a past paper question from this. So we can quickly move on to dengue and other things. We did this one, right? Did we do question number 1? No? Okay. Do question number 1. This is from methods of preservation. I told you, I think there are 3 methods. You prevent entry. Then what do? You prevent their growth and activity. Then you kill. No? There are 3 principles of food preservation. Prevent entry, reduce the activity of them or grow, or you can finally kill them. Right? So we will do question number 1. Dengue immunity of ornamental fish genome project, monthly vegetative propagation, uh, 3 principles, nanoshells. Yeah, just one MCQ is there. Taggalakanban, question number 1. Okay, online people, send me the answer. What's the answer? Yes. 5. Okay. Did you see where I? Okay. Destroying microorganisms. So, what are the ways that you can destroy microorganisms? Yeah. Drying, usually we try to prevent the growth. Ne? Use of chemicals can kill. Yes, use of certain chemicals can kill. Chilling also reducing the activity. Smoking also reducing activity, so you can't use. Salting, what's going to salt going to do? Again, by removing the water, yes, you can uh, stop the activity, reduce the activity. And use of chemicals can kill, salting can't. Freezing reduces activity, sugaring is also reducing activity. Yes. Radiation and heat treatment. By heating, you can remove. Yes. By destroy, you can destroy. Ne? Online. Smoking. Yeah, yeah, it has antibacterial properties. No, yes, yes. Smoking has antibacterial properties. Yes. Smoking you can take, but chilling is something done to just to delay also. Ne? Chilling, you are just delaying things. Yeah. Any questions? This was done last week. No. Okay. So we can move on to dengue. Right. Few MCQs. This year the MCQ could be increased because we are facing a massive dengue pandemic, right? So they have a trend of asking those in that way. So there could be a dengue MCQ. So let's see. Don't focus target on those things, but good to know these things, and it's just some general knowledge you need to have. So it's a waste not to study these things. Sorry. Okay. Dengue and filaria. First, we'll talk about dengue. Then a few things. It's caused by an RNA virus. Okay, dengue is caused by an RNA virus. The family of arbovirus group. Okay, arbovirus. It's an RNA virus. So that's why you can have so many varieties of dengue. One person who got dengue a person who got dengue once can get infected with a different variant of dengue. Okay, that is because why? It's an RNA virus. RNAs mutate very fast. So that's why. So you might think, okay, a person they got the dengue virus one time, they can have what? Immunological memory. Yes, that happens. Immunological memory is there, but usually, if you get dengue for the second time, that is going to be much serious than the first time. That is because your immunity is already weakened, and you are going to get infected with a mutated version that is usually more virulent, okay, because it is a RNA virus. That's why again and recurrent infections of dengue is much more dangerous. Tropical and subtropical regions, okay. Two species you need to remember with the spellings. Aedes aegypti. So Aedes, A and E is used right, very generously, and Albopictus. Those are the two mosquitoes. They can ask in the structured essay to write. There are abiotic factors and abiotic factors. Biotic factors are what? The virus and the vector and the host. Those are the living. So virus, we say that is related to the biology. Okay, so the vector is the mosquito. Imagine the host is human. The abiotic factors are what? The factors affecting the breeding and the growth of the vector. These are actually vector related abiotic factors. So the temperature for the vector, humidity that is good for the vector, and also rainfall. Got it? Is transmitted to human by biting of an infected female mosquito. This is some things you know from the Chutikari. Okay, the infected humans could be symptomatic as well as asymptomatic, and they are the carriers and multipliers. The virus only multiplies inside humans. Okay, virus multiplies in humans. That's why they are called as multipliers of the virus. So a person having dengue is the most dangerous one because a mosquito is not responsible. For the mosquito just randomly bites a dengue infected person. Then from that person, he is going to transmit to another person. So who is actually the carrier? It's the dengue person with dengue. The dengue mosquito is like unknowingly he is going to bite someone and transmit to the next person. Okay, then you don't need much of the description. Small to medium sized. Remember, they are sort of dark in color. Okay, white markings, bands on the body. You know these things, and the arrangement of bands is different on the body and legs. They live for about 2 to 4 weeks. All right, I have given you a dengue mosquito. Can you see if you can identify those features in this mosquito? Can you see these bands on the body and the legs? Yes. In the tute, can you write down the features of the dengue mosquito? Can you write down the features of the dengue mosquito? What are the features of the dengue mosquito? Can you write down near the diagram? Yeah. White bands you can see in the body, and it's a dark mosquito. Then I want you to write down the two names of the two dengue mosquitoes. Pay attention to the spelling. Aedes aegypti. Yes, it's a scientific name. Underline, start with a capital, that's all. Okay, Aedes aegypti. Aedes albopictus, right? Those can be asked to write in the structured essay paper. Albopictus. Okay, write down those two names. Okay, then there's a bit of life cycle of the dengue mosquito. All right, so in the resource book, can you read the life cycle has 4 stages? Yes, life cycle has 4 stages. You have the eggs, then the eggs are going to become a larva. Larva is going to become a pupa. Pupa is going to become the adult. So just if you can highlight the stages, that's good enough. Okay, then the female mosquitoes lays eggs singly on the inner surface of wet containers at the water level, preferably clear water. Preferably clear water. So clear, can you write down here? Clear water, singly. Okay, eggs laid singly. Okay, inner surface of containers. So we say the eggs are sort of slightly resting on the edge. Maybe if you have the sort of maybe a yogurt cup or something, these dark color eggs are sort of resting on the edges, sort of half floating. Okay, they are resting on the edges. Okay. Then initially the laid eggs are white in color. They become shiny black. Can you highlight? Shiny black. The eggs become shiny black within a few minutes. The eggs are smooth, long, and ovoid. Okay. The eggs are smooth, long, an oval shape, ovoid. Okay, 1 millimeter long. The eggs hatch within 2 days, within 48 hours. Can you see? Can you highlight this? The eggs hatch within 48 hours. Highlight this to become the larva. Okay. The larvae rest with an angle to the water surface. They also sort of they in this water container they are also sort of they are sort of lying with their back resting on the sides of the container. Right? They don't move much. The larvae don't move much. Okay. The eggs can undergo a dormant period for about 6 months. That is the most dangerous thing, right? Eggs can be dormant up to 6 months. So imagine in this rainy season, a female mosquito laid eggs, but somehow you destroyed or overturned, or somehow the eggs were not exposed to enough moisture this rainy season. But in the next rainy season, within 6 months, if you get rain, right, they can become again, they can hatch again. Okay, that is a danger. Then the body of the larva consists of three parts. Can you label the three parts, Methana? Right? It's not very big. Okay. Body of the larva contains three parts. There's a head. Then this is the thorax. Okay. These all are abdomen. Head, thorax, and abdomen. Then after 4 to 5 days, within 5 days, it's going to become a pupa. Pupa is coma shaped. Coma shaped. Okay, and mobile. Can you write down? Coma shaped and mobile. Those are the people that you see. Right? If you take a look at one of these water containers, you might see they are dancing around. No, have you seen this? Those are the pupa, right? Mosquito pupa, because the larva is not mobile. The larva is sort of resting on the side of the container. The pupa is the one that is moving everywhere, very mobile. Pupa becomes a mosquito within 1 to 2 days. So you can write here, within 1 to 2 days, there's a pupa coming out. Sorry, adult mosquito coming from the pupa. Okay. So that's the short note I want you to have. You have to be able to describe about the eggs, how the eggs are laid, what is the color of the legs, eggs, those things. Enough. Okay. Then here, what's going to happen? Okay, read the resource book. Breeding sites. Female mosquito lays eggs in a wide variety of artificial and natural wet containers. They prefer dark color surfaces with clear, unpolluted water. Okay, so dengue mosquito breeding site, right? It's very particular. You will see this when we compare the dengue mosquito breeding site with Filaria mosquito breeding site. Okay, the dengue mosquito breeding site, they prefer clear, unpolluted water. They need fresh water. If you take a look at the Filaria mosquito, he prefers to breed in very dirty toilet water or these husk pits, right? Drainage pits, those places where the Filaria mosquito breeds. That's why Filaria is common in these very low sanitation areas, poor economic areas. Filaria is common. But dengue is common as soon as it rains. Fresh water is there. Okay. Breeding sites can be found in indoor as well as outdoor. So they have a general idea about few of these sites. Discarded plastic containers, such could be clay pots, yogurt and ice cream cups, bottles, cans. These are general knowledge things. Okay. Automobile tires and machinery parts. Okay. In these sort of junkyards, you can see. Okay. What else? Building structures as roof gutters and concrete slabs. That's why people are encouraged to build roofs without gutters now, so that the water directly falls into the ground. Household institution refrigerator trays. Can you highlight that? So modern fridges do not have refrigerator trays, flower vases, squatting pans of washrooms. Okay, so if the washroom is not used for a long time, then mosquitoes can breed in the water of the toilet. Okay, I mean like that water seal area, it has fresh water, no? So mosquitoes can breed because can you understand? Within up to within 2 days they become the larva. Within 5 days they become the pupa. Overall, yes, 2 days, 7 days, maximum of 9 days is enough if you have stagnant water, but it can happen in 8 days now. From eggs to adult, 8 days is enough to go from the eggs to the adult. Okay. Natural breeding sites. So, in sort of small certain decorative plants, you might have seen small amounts of water in between leaves collected. Right. That is enough. That is leaf axils. Okay. So those things are general knowledge. We better move on to the stages of dengue. So you can see uh, when the mosquito bites an infected person, he is going to get the dengue virus. The mosquito is going to get. Then the virus is going to be injected into a non-infected human. That is the transmission of the disease. Okay. Right. So then we have to move on to the symptoms of the disease. So for that, I gave you these two monitoring charts. I think you can't clearly see, ne? Can you see this? Online people, I think in your own PDFs, if you can better see this. This is called the blue chart and red chart. The nurses usually call this in medical words blue chart and red chart. So this blue chart, we call it fever chart. Okay. So can you see management of dengue in adult patients without evidence of fluid leakage? Okay. So can you write down here without, yeah, just to clear it, without evidence of fluid leakage. Okay. That is the first stage of dengue. Okay. The blue chart. That is to monitor patients who are in the early stage. They are having only fever. But you monitor their fluid input and output, and also you monitor their temperature and also platelets. What are the things you can see? So the whenever a patient is suspected to have dengue, the ward intern doctor has to do so many things, right? Sometimes out of the in a ward with about up to 50-60 patients, there's only two intern doctors. Sometimes one has to be allocated just for the dengue patients. Okay, so out of the two, one is doing only dengue patients, the other one is doing all the ward work. So here you have pulse rate, blood pressure is very important. You will see. Then, okay, this is capillary refill time. That is something we need to measure. Okay, then you have respiratory rate here. PCV's packed cell volume, that is something we check to see if the person is losing blood plasma. Then you have platelet here, very important one, platelets and white blood cells, WBC. Those are the things that we have to mark in the blue chart. So usually I think every hourly, right? Every hour you have to monitor in the blue chart. Okay. Then there's the red chart. So we'll read the resource and see what is going to happen before we move on to the red chart. Okay. So in the blue chart is for the early stages of dengue when we are suspecting dengue fever. Let's read online people what's going to happen. Okay. Dengue can affect infants, young children, and adults. It starts with flu like symptoms. Sudden onset of high fever, highlight. Okay, sudden onset of high fever is the first thing. Then, what are the other symptoms? So, nowadays the symptoms are very vague, right? There could be a wide range of symptoms. Okay, what are things? Usually, headache, pain behind the eyes, very unique thing. Okay, highlight that pain behind the eyes. Muscle and joint pain, very common. Nausea, they vomit a lot. Okay. Skin rash, something that happens later. Okay. Skin rash doesn't happen early. Symptoms usually last for 2 to 7 days after incubation of 4 to 10 days from the bite of an infected female Aedes mosquito. Okay, you can draw a small timeline for the progression of dengue fever. Okay, progression of dengue fever, you can draw a small timeline. Here is the bite. You get bitten by a dengue mosquito. Then, for about 4 to 10 days, the disease is silent because the virus is multiplying in your body. For about 4 to 10 days, the disease is silent. So, this is the biting point. Then you have about 4 to 10 days, what we call as incubation. That means the virus is multiplying. 4 to 10 days of incubation period. Then you enter into what we call as the febrile phase. Febrile phase is the thing that is you are having fever and this pain behind the eyes, joint pain, everything is there. Could be up to 2 to 7 days. 2 to 7 days. Okay, 2 to 7 days. You can have these early symptoms. That is the phase that we monitor using this blue chart. Understood? You don't need to know the name. That is called the febrile phase, but you don't need that. Okay, so here you are going to have mainly fever and the remaining body ache, joint pain, those kind of things, headache, nausea, vomiting. Okay, then you are going to move on to the severe part. Okay, can you keep on reading? Severe dengue or dengue hemorrhagic fever is a potentially deadly complication due to blood plasma leaking. Okay, the signs could occur, so it is kind of overlapping. Either remember, it could occur in the middle of the this febrile phase. So keep in mind, a person doesn't have to go into this phase that we are talking about. So he can go in this good pathway, right? That is, he can recover, right? After 2 to 7 days of fever, he can recover. Recovery. Okay, that is one option. So that is good if you went in that pathway. Then what could happen if you don't heal within 2 to 7 days? You can enter into the dangerous one that is called leaking phase or dengue hemorrhagic fever. Dengue hemorrhagic fever. That is the dangerous one. Therefore, we start monitoring a patient who is going to show these symptoms. Okay, listen carefully. Okay, blood plasma leaking could be there, fluid accumulation, respiratory distress, and severe bleeding, organ impairment. Okay, warning signs occur 3 to 7 days after first symptoms in conjunction with you are going to the fever is going to go a bit normal. Okay, in the dengue hemorrhagic fever, the fever is going to drop a bit. Abdominal pain you are going to get. Okay, I'll explain why. Persistent vomiting, you can't stop the vomiting. Rapid breathing, you are going to get bleeding from nose and gums. Fatigue, impairment of liver, reduced number of platelets, blood in vomit. Okay, so what would happen? The next 48 to 24 to 48 hours could be lethal. You can go progress into massive bleeding, very low blood pressure, shock, and death. Okay, so hemorrhagic fever would progress further into what we call as dengue hemorrhagic shock. Right? Within 24 to 48 hours, you can progress into shock. Shock එකට ගියා කියලා කියන්නේ it is very unlikely he will recover. From hemorrhagic fever, there could be recovery as well. From shock, the recovery is a bit unlikely. Shock will most likely progress into death. Okay, shock will most likely progress into death. This is the timeline. You get bitten by a mosquito, there's 4 to 10 incubation period. 2 to 7 days you can have fever. Within that 2 to 7 days, after about 3 to 7 days, right? After 3 days of having fever, you can enter into hemorrhagic fever. After developing hemorrhagic fever, keep this timeline in mind. Within 24 to 48 hours, you can go into shock. Okay, so you need to remember in what every stage what is happening. In hemorrhagic fever, what do you get? In hemorrhagic fever, what do you get? Can you write down here some of the things? You're going to get abdominal pain, right? Make a small note, online people. You're going to get abdominal pain. Then persistent vomiting. You can't stop the vomiting. Okay, that means the person is in hemorrhagic fever. Sometimes there could be blood. Okay, in the vomit there could be blood. Okay, then bleeding from gums and nose. What is that? Why is that happening? Because of the damage caused by the virus, your platelet count is dropping. The platelet count dropping means what? That means your bleeding, your coagulation is not happening properly. So what's happening is you are, and also your blood vessel permeability has increased. From your capillaries, you are losing the liquid part of blood. Blood plasma is getting lost. Okay, so therefore your clotting factors are being lost, therefore blood is not clotting, so you are going to start bleeding from all these sides. Okay, you are going to get enlargement of liver due to the leakage of plasma inside the liver. Blood plasma is getting collected. Okay, understood. Then if you enter into the shock stage, what would happen? You are going to get very low BP, very low blood pressure. You're going to get very low blood pressure, and then massive bleeding. Okay, massive bleeding. You can have blood in your stools. That means from the in the stools you can have. Okay, and in the vomit, of course, you're going to increase the amount of blood you're going to have in vomit. And then finally, because of the massive loss of blood and blood volume, the circulation process is going to be impaired and the person is going to die. This is the timeline of progression of dengue. Okay, quickly take this down. You can add your own points by referring the resource book. Okay, okay, right. So that is have a small idea. Can we save the person when they're on the last stage? You can give them platelet, right? You can give them a pool platelets, but to prevent the bleeding, then you can give them crystalloids and try to increase their blood volume somehow. But at this point, even in the ICU, at the shock stage, it's like a at the shock stage, imagine you are trying to fill water into a container with a lot of holes. Everywhere there are holes because all the blood vessels have become leaky, and you can't stop the bleeding because all the clotting factors are all gone, have leaked out with the blood plasma. So there are no clotting factors to prevent the bleeding, and also blood is the liquid part of blood plasma is also leaking. So whatever you put inside your body from the veins or blood or whatever, they are also going to go out. Okay, so there is a problem with this. Got it? Clear? Okay. No, you can't do yet. So we'll finish talking about dengue. What are the controlling measures? What are the controlling measures that you can take to control the dengue vector? These are general things. Dengue vector controlling methods are mainly aimed at both immature and adult stage of the Aedes mosquito. Okay. Then you can categorize your breeding methods. Sorry, controlling breeding. You can categorize them into. Okay. That's the rest of my ISO. Controlling measures. Okay. So you can have environmental methods, chemical methods, as well as biological methods. Okay. You can highlight this very important. The most effective way they can ask in the structured essay paper. What is the most effective way? Most effective way is controlling the vector by eliminating its breeding sites. That is the cheapest. Okay. Thing to do is to prevent the vector from multiplying. Is to prevent the vector from multiplying. That is the cheapest thing to do. Okay, that is the cheapest thing to do. Is to prevent the vector from multiplying. Then you have to remove how to remove the vector from how to stop the vector from multiplying. You remove sources of water, right? Fresh water accumulated places you can remove. That's what you need to do, right? Few things you need to remember, okay? Because this is applied biology. In applied biology, we have to know about how to apply your biological knowledge to solve a problem. What is the problem? The dengue spread. Okay, so having continuous water supply to minimize storage of water in cement tanks, barrels, and other containers. That means better if you can have a tap line rather than you are collecting water in tanks and tanks. Continuous flowing water. Okay, flowing water. Then this word is important. Mosquito proof water storage cement tanks, domestic wells. You can cover them by cover them with a net. Okay, you can cover them with a net. Buildings without roof gutters. In these modern buildings, have you seen they don't have gutters? The water just falls onto the ground where there is maybe a drainage part so that the water can flow away. Okay. Removal of unserviceable roof gutters. If you can't clean the gutters, better to remove them. Better to remove them rather than having gutters that you can't clean. Okay. Then regular cleaning with scrubbing of water tanks. Why do you need to scrub? This is a key word. Why do you need to scrub? Right? Whenever you are writing this answer, I want you to write scrub. Can anyone tell me what do you need to scrub? Yeah. To remove the eggs that are sticking. Because why? The eggs have a dormancy period. Ne? Right? They can remain dormant for up to 6 months. So just because you removed all the water once and you let it dry doesn't mean you kill the mosquito. You have to scrub it and clean because the next time you fill water into it, the eggs are going to become activated. Proper disposal of solid waste such as your rubber tires and your yogurt cups and ice cream cups and everything. Yes, Medini? Tires and household and garden utensils, right? Flower vases and other things. You have to properly store them. Okay. Then these are environmental management. So you can put this sort of a thing and you can say these are environmental methods. You can manipulate your environment so that the breeding sites are reduced. Understood? You can manipulate your environment. Then you can have biological controlling methods. Those are not very widely available. Okay. So they are done to control the vector. Two things they are given in your syllabus. That is, you can have fish species that feed on the larval stages. Can you highlight? Just have a small idea. Gappi දාන්න පුළුවන්, දණ්ඩි දාන්න පුළුවන්, tilapia දාන්න පුළුවන්. Okay, no need to remember the scientific names. Okay, just remember Gappi. Okay, if they ask, can you name a fish species that can be used to control dengue? Just write Gappi. Okay, just write Gappi. Okay. Then, then there's another biological controlling methods, that is a bacterial right? That is a bacterial methods, right? Bacillus thuringiensis israelensis. Can you understand? So Bacillus thuringiensis is the scientific name. Ne? So what do you mean by israelensis? That is a subspecies. Yes, you remember this. Ne? So it's a subspecies of BT, Bacillus thuringiensis, that can be used to control. Right? It produces endotoxin. Can you highlight? What do you mean by endotoxin? Now you need to know these things. We have done microbiology. What do you mean by endotoxin? It's a part of the bacterial cell wall. Yeah. So it is produced, and when the bacterium dies, the endotoxin will be released. Okay. So highlight that, very important one, Bt toxin. We have done this. I think in Sri Lanka, we tried sort of spraying Bt bacteria into waterways so that they can multiply and produce this toxin when they die. So that toxin in the water, that means they can kill the larval stages. Thermal fogging, that is a chemical method, I think. Yes, you take thermal fogging as a chemical method. Okay. So you need to know what are the environmental methods that is given here. Then what are the biological methods? That is two things. Using fish that eats the larval stages, and also using Bt bacteria, okay, which produces the Bt toxin. The chemical method is fogging. For dengue mosquitoes, right? They should be exposed in sufficient dosage, right? They will be knocked down and killed. There are several limitations in controlling methods. Okay. Then, in biological controlling methods, particularly, there are several weak points. Okay. When fish are used, right? These are general knowledge things. The fish could die due to the lack of food. Okay. And due to the changes in water quality parameters, you know these things of pH of water, right? You can't apply fish in everywhere, right? Chuti yogurt cup you can't put a fish, no, right? That is not practical. So depending on the place, you have to adapt to which method is best. Then, if the domestic water containers contain chlorinated water, if you put fish into like a concrete tank containing chlorine water, they may die. Okay. But BTI, then what are the limitations for the BTI toxin? Bacillus thuringiensis israelensis, BTI, right? There could be some breeding sites where BTI can't be applied because BTI is a bacterium, so you can't apply him to drinking water sources, right? Water sources that we would take for drinking, so you can't apply. Then chemical methods, of course, you know fogging. No? What are the problems with fogging? It's very irritating. Ne? You can't stay in the area that has been fogged. Some people can have reactions such as asthma reactions, allergy reactions, because we use a mixture of kerosene and other chemicals. Kerosene uddana, kerosene oil we used to sort of burn so that we get the fogging. Okay, so the fog. So remember these limitations. These are the limitations that we are going to face when we are trying to control the vector. That is all about dengue. Clear? I think we had a question from dengue. Did we have? Oh, no. That is asking about both dengue and filaria vector. Yeah, okay. Then we'll come to file area. We'll come to file area. Okay, so we'll go through the resource book first, then we'll see what diagrams we have to understand. Okay, uh, filaria is one of the oldest debilitating disease. Debilitating disease means it's going to disable you, disfigure you, and it's going to be sort of it's a disfiguring disease. You might not be killed from it, but it's going to make you deformed. That's one of the things interesting things about filaria. You might very unlikely that you might die of filaria, but you are going to live in a deformed, uh, in a diseased state. It's one of the major leading cause of permanent and long-term disability. Right? It does not kill people. That is interesting. Right? You don't usually die, but causes permanent disability. It's mosquito borne by there's a nematode, that is a worm. Filaria worm is the main. I mean, that is the pathogen. Okay, and it is so the nematode actually lives in the human lymphatic system. So they are very small thread like, you know, our lymphatic vessels are very small things, right? Very small. So the worm lives inside, so it is exactly almost the thickness of your lymph vessel. So if these are your very small lymph vessels, the worm nicely fits inside this lymph vessel, completely blocking it. So Wuchereria bancrofti, Brugia malaigera, there are two main filaria worms, nematodes. Okay. It is endemic in developing countries with poor socio-economic. Can you highlight that? It's associated with poor environmental sanitation. Right. So you need to know only Wuchereria bancrofti. Please highlight the nematode name. There can be question in the structure. They say who is the parasite of filaria. Practice the writing the name Wuchereria bancrofti. Wuchereria bancrofti. Okay. Then who is the vector? So 90%. There's another one called Brugia malai. You don't need to know him. Wuchereria is enough. In Sri Lanka, the mosquito is Culex. Quinque fasciatus. Quinque fasciatus. Okay. Remember the spelling. Breathes in polluted water. Highlight blocked drains, toilet pits, husk pits. Husk pits, I told you. When did I teach you about husk pits? In industrial microbiology, when you're talking about extracting fibers. Yes. What is the enzyme that is produced by microorganisms to extract fibers? Happening in husk pits. Online people, what needs to be broken down if you want to separate the fibers? What is the cement between plant cells? Yes. What is the cement between plant cells? Pectin. So if you want to break down pectin, what do you need? Pectinase. Yes. So in husk pits, there's a lot of pollution, right? So you don't need that here, right? In husk pits, there's a lot of pollution. So the Culex mosquito likes to breed in that polluted water. That's why it happens mostly associated with very low sanitation conditions. Okay. Then the mosquito, small to medium size, gray black in color. The nervous, that is another name for the sort of the edges of the wings, okay, beset with brown or blackish scales, and the posterior margin of the wings are fringed with bristles and scales. Okay, so let's take a look at the mosquito. Let's take a look at the mosquito. So this is the filaria mosquito. Please write down the features. It's sort of gray color, right? Gray color. Okay. You can have bristles and scales. It's not exactly visible here, but you can write down here. Okay. Write down posterior margins of the wings. Posterior margins of wings fringed with bristles and scales. Quickly write down. Fringed with bristles and scales. Okay, online people. Okay. So the features of mosquito are not that important. Then okay, let's come to the life cycle. It's a bit blurry. Okay. So then not very clear. No, is it blurry in your the PDF I sent you as well, or is it? I think I downloaded the proper CDC photo. Oh, now it's better. Okay. Okay, so the life cycle of the mosquito is like this. Okay, the life cycle, there's a mosquito stages. Okay, the life cycle of the mosquito is like this. So let's read what is in the resource. Females lay eggs in the form of egg rafts that float on the surface of water. Right? Females lay egg rafts. Okay, lays eggs in egg rafts. Rafts means what? They sort of like this. They float on the water, right? They float on the surface of water. All the eggs are like sticking together. Okay. After hatching, the larva rests, keeping its body at an angle to the water surface. That is kind of similar to the dengue larva. Okay. Then factors associated with transmission. Transmission in a community is influenced by number of infected people. So you can see this is progression of lymphedema. We'll come there later. Okay. Density of microfilaria in the blood of infected people. I'll explain to you what is microfilaria. Okay. Then density of vector mosquitoes, that is your Culex quinquefasciatus. Yes. We didn't write down the name of him. Ne? Who is the name of him? Culex quinquefasciatus. Okay. Practice writing that name at least once or twice so that you can write it in the paper. The mosquito is Culex conquifasciatus. Okay. Okay, so the methods of transmission you can see here. What would happen? It's a bit blurry, no? Is it blurry in this diagram I see? PDF. Okay. Does it become blurry? Yeah, kind of, no? Yeah. But you can slide of kind of lead it, ne? Yes. I don't know why. Okay. So we'll go along with the order that is given in the resource. Okay. Filaria parasite is introduced to the human body by a bite of a female adult mosquito. When the mosquitoes suck blood from humans, the larval state of the parasite falls onto the skin. That is very interesting. Can you see here? The parasite, what we call as microfilaria, right? So the microfilaria is a larval stage of the filaria nematode. You don't introduce the mosquito doesn't directly introduce him like the dengue virus. The microfilaria falls onto the skin of the person, and then what happens? They penetrate through the skin by the, I mean, through the mosquito's bite wound. So when the mosquito is biting you, he creates a wound. He put his sort of the blood sucking proboscis into the skin, and there's a small hole. Through that hole, the microfilaria enters. Can you understand? Highlight the larval stage of the parasite falls onto the skin, then they penetrate through the bite wound and enter the human vascular system, blood circulatory system. From there, they migrate into the your lymphatic system. Can you see? So the microfilaria are found in the lymphatic system, migrate into lymphatic and peripheral blood circulation. Okay, we'll write down those things. Okay. Within 1 year, the larvae become matured into adult worms, mainly the adults. These are the adult worms. So this is the smaller one is the male, the larger one is the female. Okay, two worms are they are living in the lymphatics. Adults are in lymphatics. Adult male and female worms nest and mate in lymphatic vessels, produce a large number of tiny eggs which become microfilaria. So from the adults you get microfilaria, which are these. We get microfilaria. They are the infective stages as well. So they are the people who are going to drill. I mean, so they are going to be dropped onto the skin and they are going to enter as well. They are going to become adults. Okay, got it. Microfilaria live in the lungs during daytime. They have a very nice uh, daily routine. Okay, during daytime, okay, microfilaria live in the lungs. Okay, at night they come into your sort of peripheral blood. Peripheral blood means sort of there are small blood vessels that is going through your skin. So that is the only way that we can diagnose. I mean, from early stages, if a person has filaria. So what do you need to do? What you need to do is you can't like cut open someone's leg and see if there are adult worms there, no? Right? You can't cut open the lymphatic vessels in a person who is not having any symptoms. What do? You have to wait until it is night time so that the microfilaria come into the peripheral blood. So at night you are going to take some blood from your finger, okay, finger prick at night. Then if you observe them under the microscope, you will be able to see the microfilaria in the microscope. Okay. So at night, what else also can happen? At night, this microfilaria can enter into the mosquito as well. At night, if a mosquito bites a filaria, having a person with filaria, the microfilaria enters into the mosquito as well. Okay. Can you see? Ingested microfilaria transform into several larval forms. Maybe in L1 then there's a L3. So several larval forms are there inside the mosquito as well. Okay. So this microfilaria undergo several larval stages into, inside the mosquito as well. Then again with another bite enter into the human body. So who is actually entering into your body? That is the L3 larva. L3 කියන larval stage එකේ Filaria worm enter into your body. Then he will become an adult. Adult will produce microfilaria. Microfilaria are in the blood, peripheral blood at night. Another mosquito bites you at night. Microfilaria enter into the mosquito. Inside the mosquito, they become L1, L2, L3. So many stages are there. L3, when you bite another person, the mosquito bites another person, L3 falls onto the skin. Then L3 will enter through the bite wound of the mosquito. Then it again will become adults. Adults, keep this in mind. Put a star mark and you can write. Adults only live in lymphatics. They don't move into blood. Adults only live in the lymphatic system. Who is going to move into the peripheral blood circulation at night? It is microfilaria. And microfilaria have there's two places where they spend their day. During daytime, they are in your lungs. Night time, they are in your blood. Can you imagine having these so many small worms traveling inside everywhere in your body? Right? That is the microfilaria traveling everywhere. And also you are going to have adults in your, can you see the progression of the disease? So this person's lymphatic system is having these worms. Okay, that's why they are having these swellings. And throughout their body, the microfilaria is circulating at night. Okay, small worms are circulating. Okay. Adult worm lives in the lymphatic system for 5 to 6 years. Can you highlight that? He has a lifespan of 5 to 6 years, adults. Microfilaria can live up to 1 year. When adult worms block lymphatic vessels, they get distorted and the lymph flow does not occur. Lymph tends to accumulate in dependent parts of the body. No, can you see? Lymph gets accumulated. So, can you see gradually there is going to be swelling. I think swelling increases during the day because why? The person is walking and doing some work. All the fluid is, lymph is at the bottom of the body, no? So their legs are swelling during the day, then later disappears at night. Second stage, it does not disappear at night. Then you are going to get bigger, and later the skin is going to get damaged, and then the skin is going to heal, and it's going to get damaged. Yes, like that, it's going to be a cycle of damage. Can you see? Can you highlight? Lymph accumulates in the legs, hands, testes. Sometimes even in the scrotum, the lymph gets accumulated and it gets bigger. Penis and the breast. Right? Breast can become enlarged due to lymphedema as well. When the microfilaria is in the lungs, right? When the microfilaria is in the lungs, you can have lung-related symptoms. You can have a long-term cough, a chronic cough. Imagine you are having worms in your lungs, right? And wheezing. Dyspnea, you can write. Dyspnea is something we call difficulty in breathing. Okay, it's a word for difficulty in breathing. You can write down here. Can you draw an arrow to this microfilaria? Can you draw an arrow and say during daytime they are in the lungs, during night time they are in peripheral blood. Okay, so at the daytime when they are in the lungs, you can have chronic cough, wheezing, dyspnea, fever. Your chest can hurt, chest pain, lungs are hurting, and weight loss. Okay, weight loss. Because the person is having worms, so that there could be a small blood loss as well. The worms are microfilaria. So please categorize these microfilaria. They are in daytime in the lung, nighttime in peripheral blood. They are in peripheral blood at night time. That's why you can identify them only at night. Okay, otherwise you can't take them out of the lungs, no? Methane. Clinical manifestations vary from asymptomatic to chronic. So this is a person that is mostly asymptomatic. Ne? Small amount of swelling is there, right? You think it's something maybe a varicose veins or he's been standing for a long time or something asymptomatic. Chronic lymphedema, this is something close to. So at this stage also elephantiasis is there. Elephantiasis means what? Your legs look like elephant legs, right? The L3 larva develops into an adult worm, remains in lymphatics for several years, producing microfilaria. So you know how much is their age? 5 to 6 years they can live. Okay. These patients are asymptomatic for 5 to 6 years. Okay. Can be only be detected through night active surveillance. Presence of adult parasite in the lymphatic system for a longer period gives rise to late manifestations. Okay, they are due to dilation of lymphatic vessels, highlight, followed by their dysfunction. So the when you have a worm inside, the lymph vessel is going to become bigger. Yes, accumulation of fluid in tissues and increase of infection. Because your feet are swollen, they can get damaged. No, you can hit them in the certain places. You can sometimes drag them along the ground unknowingly. Uh, you can damage these swollen organs. Okay, so you can have skin infections. So can you see this happens because of the recurrent skin infections, right? Your skin is getting bacterial fungal infections, so that there's a layer and layer of damaged skin. With recurrent infections, skin of edematous extreme become thickened, gives rise to warty nodular papillomatous appearance. So warts again, right? Lymphedema is graded according to the state of edema and the appearance of the skin. Can you see? These are the stages. Up to from stage 1 to stage 7, you can have lymphedema stages. I just gave them for just your knowledge. Elephantiasis is not the worst stage, right? So, stage 7 is the worst. I mean, you can have elephantiasis from stage 4 onwards. Okay, got it? Can you see? Clear? Then we move on to controlling measures. Controlling measures. What can you do to control? Okay. Right. Controlling measures. Personal protection from mosquito bites and prevent mosquito bites by the use of mosquito nets, repellents, long sleeve shirts. Obviously, those are ways you can prevent mosquito bites. Highlight uh, mosquito nets, repellents, long sleeve shirts, trousers. Elimination of breeding sites. Repair broken septic tanks, clean drainages, preventing dumping garbage so that when you dump garbage, the drainage just get blocked. Yeah. Chemical control of aquatic plants in water reservoirs. Yes. Have you seen this salvinia? Okay, when they are overgrown in this water reservoirs, the water becomes polluted, right? So that is a nice place for the culex Kinky fasciitis. Prevent creation. Use of larvivorous fish. Can you highlight larvivorous fish? Such as guppy, nalahandaya. I don't know who that is. Right? Guppy matak tiyagandu. Okay. Screening healthy population. Yes. Very, very important. Night blood films. Can you highlight the word night blood films? Right? For people who harbor the infection but do not show any symptoms. That is very important. Put a arrow there, right? It's a very important thing that we do. Have you ever had PHIs come to your home at night to collect, get a few drops of blood into microscope slides? No, right? That happens in an area. If they identify a few patients with filaria, then they will cover the entire sort of the that division. Okay, night blood films. Why is it done at night? To detect, right? Night blood films are done to detect microfilariae in peripheral blood. When you observe them under the microscope, you will be able to see this. The microfilaria will be able to see. Monitor vector mosquito population for density. So these entomologists, what they can do, entomologists are the people who study insects. No? What they can do is they can randomly catch some mosquitoes, okay, and they can see, okay, if there are Culex quinquefasciatus among these. So if you have Culex quinquefasciatus, there's a risk he might be spreading filaria. Okay, so those are the controlling measures. Any questions about filaria? Any questions about filaria? There's a all right, let's discuss this as a question. MCQ number 2, Taggalaganba. Quickly do this MCQ, online people. Yeah, what do you think is here? Okay. The question, transmission of filariasis within a community, frequency of human vector contact. What do you think? Yes, that is important, no? Because from the vector, when the vector bites, the larval stage of the filaria, the nematode falls, usually the L3 one falls onto the skin and then enters into the body. Density of microfilaria in infected mosquito. Usually you don't have microfilaria in infected mosquito. You have other larval stages. Okay. Can you take it? Is it one of the factors? No, right? Density of no, yeah, we don't check it. Okay, in the vector, you don't usually detect it. Number of infected ones, of course. Characteristics of the vector, you can, right? ACD, no? ACD. Okay. Right. What about this one? Uh, Following could be used to control dengue as well as dengue vector as well as the filaria vector. What can you do? Question number 4. Quickly send the answer. Yep. Dengue vector as well as filaria vector. So you have to have an idea about the preferred breeding sites of the dengue vector as well as the filaria mosquito. Yeah. So let's see. Building without roof gutters. So in roof gutters, usually it's only dengue mosquito breeding. Ne? Filaria mosquito doesn't usually breed in roof gutters. So this is something only for dengue. Okay. Domestic wells. What do you think? In domestic wells, do you have filaria? It has to be polluted, no? Polluted water. So this is also again for dengue. Prevention of creation of vector breeding sites, that is also, it's like a very, uh, what do you say? It matches with any answer, no, right? Of course, you have to prevent breeding sites. Use of fish that feed on the mosquito larvae, yes, such as guppy, right? Larvivorous fish can be used for both. Broken septic tanks, that is only for filaria, no? Yeah, because septic tank means what? Sort of wastewater tanks, could be toilet tank also, right? Septic tank is something that is where filaria mosquito, Culex quinquefasciatus breeds. So only C and D, no? Yeah. So these are the types of questions that can be asked. Okay, we have a small three more topics to talk about. That is nanotechnology. Okay, nanotechnology. We have to talk about. I think yeah. I think small diagram I have included in the tute. Then shall we move on to talk about nanotechnology? Okay. So nanomaterials. We'll talk about these things. Okay. Come to the resource book. Is the emerging science involved in designing, building, and manipulating minute structures at a nanometer level. So you need to know what is the scale of the nanometer. What is the nail of the scanometer? Is 10 to the power minus 9 meters. Ne? Is equal to 1 nanometer. Then there's a definition. There's a definition. Please, can you write here? Nanotechnology. Kira, let's as a subtopic, we'll write down a small note. Can you highlight the definition? Is the creation and use of materials and devices on the same scale as molecules and intracellular structures, typically less than 100 nanometers. Okay, so nanotechnology, you have to remember, deals with less than 100 nanometers. Okay, so if it's above 100 nanometers in size, it is not something nano. Okay, got it? If you want to keep it nano, then it has to be less than 100 nanometers in size, whatever you are doing. Physical and chemical properties of tiny molecules are slightly different than bigger particles, as they have a high surface area to volume ratio. Can you remember this? We talked about this when we talked about microorganisms as well, no? Because they have high surface area to volume ratio compared to their body volume, their area is much high. Okay, the body surface area is much high compared to their inner volume, so that they have a larger surface area to communicate with the environment. American physicist Richard Feynman, no need to know about that. Living organisms are built of cells that are typically 100 micrometers. So can you imagine, at compared to the nano level, our body cells are massive. Yes. Our subcellular organisms are much smaller. Proteins are even smaller with a size of just 5 nanometers, which is comparable with the smallest man-made nanoparticles. Uh, okay, using nanoparticles as very small probes that would allow us to investigate cellular machinery without introducing too much interference. Okay, understanding of biological processes on a nanoscale level is a strong driving force. Nanotechnology has become a big business with applications in material manufacturing, energy, electronics, and engineering. Right. Application of nanotechnology for human health is termed nanomedicine. Can you highlight that? They can ask what is nanomedicine. They can ask what is nanomedicine. Applications of nanotechnology for improving human health is nanomedicine. Please highlight that. What do you mean by nanomedicine? Okay. Small part. I mean, we are in the very final stage. Okay. Quickly, we can finish this. Titanium dioxide and silver nanoparticles are used for sterilization of operating theaters. So under nanomedicine, we'll write down some of these applications. Nanomedicine, AgO2, Ag and TiO2, titanium dioxide. Titanium dioxide and silver for sterilization of operation theaters. Also, silver nanolotions are there. Silver nanolotions are there. Silver nanolotions are sometimes applied to prevent entry of microbes. Okay, you can have a silver nano coating. Okay, silver. And nanofilters. Titanium dioxide and silver nanofilters prevent the entrance of tiny particles such as viruses. They also can be used. Both of these, Ag and titanium dioxide, can be used to produce nanofilters. Okay, nanofilters. Particularly in these anesthesia machines. Have you seen these anesthesia machines where they give you the gas for the patient so that they can undergo surgery? So in that machine inside, you have this nano filter so that the gas they are giving to the patient is completely filtered out, because they are using the machine again and again. It's a machine costing tens of millions. So that machine has to be used for several years, so that they have a replaceable filter, so that they are giving gas to the patient to anesthetize so that we can do surgery. that air is very, very clean. Okay. Nano filters I haven't given. So here, this sort of under here, this sort of production of nanofilm sheets, you can write here nano filters are produced. Okay, nano filters. Then nano device sensors are used to monitor blood pressure, blood oxygen levels, and hormone concentrations. Okay. Nanoparticles can unclog blocked arteries and detect and eliminate cancer cells. So these nanoparticles, these are nanoparticles. Okay. So these inorganic based nanoparticles are there. Okay. So organic based nanoparticles can be there. So they can be used to unclog blocked arteries and eliminate cancer cells. Then there's another thing they can ask, what are smart drugs? They can ask, what are smart drugs? Okay. What are smart drugs? These are all very highly experimental and highly expensive procedures, even though they are in your A level syllabus. Okay. So they can be used, done using silver nanoparticles. Yeah, you can write it here. Sorry, smart drugs, you can write here. Using gold nanoparticles, you can make these smart drugs that can deliver the medication directly to the site where you have the problem. A special target cells such as cancer cells to deliver the cargo that would destroy these cells that are damaged rapidly and effectively in a silent manner. So to target special cells only, okay, so that the smart drug can go directly into this cancer and directly destroy. Because I mean, you know, when you give this chemotherapy to cancer patients, usually what we give is either we prevent we prevent mitosis, right? So by preventing mitosis, of course, we prevent the tumor from becoming bigger, but that also affects the normal body cells as well. So everywhere cell growth division is affected. So that's why the person is going to have hair loss, right? Skin defects. They're going to feel very weak, right? They are normal repair mechanism, nothing else. But if you can just target the cancer only using this delivery method, then least side effects. Then nano shells. I think I had one. Nano shells. Mm, particles. No, right? Okay. Special nanoparticles consisting of dielectric core nanoshells are used in treatment of cancer. A nanoshell is slightly bigger than a poliovirus. Gold nanoshells are used in bioimaging enhancements as well. Right. So nanoshells is the next thing you want to know. There's a question as well. Yes. State two uses of nanoshells. Quickly write down the answer for this. Treatment of cancer, and there's a special one for gold nanocells. Quickly write that down. What is the main the application for gold nanocells? Because the gold nanocell, can you understand? If you imagine, if you want to visualize a certain part of your body, for example, a blocked artery or something in the brain, you can send the gold nanoparticle to that particular area, and then when the patient is in the scanning machine, they will detect where the gold particles are, so that you can uh, easily and very precisely identify හරියටම කොහෙද where is the problem. Okay. Then, in treatment of diabetes, a nano device is attached to the body which can release the required dosage of insulin. Yeah, small nano devices that are made in the nano scale, you can implant them under the skin so that at regular time intervals they can release insulin. Okay, that you can do, because can you see this nano shells is something that is very hidden in the syllabus. They have asked it in the structure essay paper. Can you understand? Uh, there has been successful application of nanotechnology for the treatment and management of pain in both clinical and experimental studies, right? So, go nano-formulated liposomes. Meti. Okay. So, nano-formulated liposomes, you can write here to deliver drugs for pain therapy. All right, due to maybe due to a myocardial infarction, heart attack, he could be facing a massive chest pain. Okay, or due to trauma there could be a massive pain somewhere. Okay, so and after surgery, right, in the surgical site, if you had a massive orthopedic surgery, imagine you had rods inserted into your bones, yes, to repair a broken bone. So you can send a liposome directly to the pain of the site of the damage so that you can deliver painkillers. Nanocarrier systems can be easily transferred to the airways. Many respiratory diseases can be treated using nanotechnology. Yes. Then Vivagel. Vivagel is a vaginally applied microbicide. Very important, right? So we'll write down about Vivagel. It's a trademark, I think. Okay, even though it is in your resource book. uh, Vaginally applied microbicide. Okay, vaginal microbicide for prevention of what? HIV and herpes simplex. Okay, nanocomposites are used to replace broken bone and to fill teeth. Right? Nanocomposites. Methi, can you see? So their composite means it's like if you take a concrete, you don't have only one thing. No, you have sort of gravel, sand, cement, and everything is there. So whenever you are repairing a bone or filling a teeth with something, you need to have like a nice concrete like material. So can you make draw an arrow? Okay, nanocomposites replace broken bones. Replace broken bones and to fill teeth, right? So using this diagram, you can add the other things. Okay, so these are the things that we have mentioned in the resource. So using this nanomaterial classification, you can sort of have a visual idea, right? Nanofilters using this sort of nanomaterial, and using organic based nanomaterial, you can have pain relief, you can have smart drugs, you can have gold nanoparticles. Uh, we could add here an imaging, the gold nanoparticles, imaging, enhancement. Yes, can you add that? For imaging enhancements, you can have gold nanoparticles. Okay. Okay, that is the end of nanotechnology. Any questions? Yeah, we didn't do this one, no? What are known as nanoshells? What are known as nanoshells? You can write down what are known spherical nanoparticles. Spherical nanoparticles with a dielectric core. Dielectric core means what? Sort of a semiconductor, no? Dielectric core. So those are the two things you have to mention. They are spherical with a dielectric core. Those two things, if you have mentioned, you can get the marks. Okay. All right. We have to then talk about stem cell therapy. Okay, stem cell therapy we have to talk about because stem cell therapy has been not much question, but applications of stem cells has been asked as a, I mean, short note question. But let's see what would happen this year. Okay, so anyway, we would have covered it, no? So nothing to be worried about. So stem cells. Before stem cells, we need to finish the human genome project. Ne? So for the human genome project, I had this yes HGB timeline. Okay, let's come to the human genome project. This is how the human genome project was conducted. Okay, I'm just showing you so that I can explain it easier. Okay, so let's see. 1990 we can follow what happened in the human genome project until finishing of the human genome project. Okay, so this is just a visual guide for that, so that I can explain what is in the resource. All right, come to the resource book. Yeah, we have to finish stem cells, but we will finish genome project and come to stem cells, right? Online people, can you come to page number 64, human genome project? Right. Human genome has been a focus of biological research for the last 2 decades and will continue to be the center of attention for many years to come. It is a worldwide effort to identify all the human genes. Can you highlight? It's a worldwide effort to identify all the human genes of each chromosome with several other goals. Okay. So, it was originally a 13-year project from 1990, coordinated by the Department of US Department of Energy, maybe, right? It was mainly coordinated by uh, US Department of Energy, DOE, right? US Department of Energy, as well as the National Institute of Health, NIH and DOE. Then additional contributions came from UK, Japan, Germany, France, okay, those kind of other countries. The aims of the project were to identify all the genes. How many genes do you have? Highlight the number, around 20,000 genes. Okay, highlight the number, 20,000 genes are found. Determine all the sequence of 13 billion chemical pairs. So you have pairs, 13 billion pairs are there. Okay, you have 13 billion base pairs. Okay, all right, let's see. Then store information in databases, improve tools for data analysis, transfer related technologies to the private sector, then address the ethical, legal, and social issues. Because why? Some people in their cultures believe our genome or our cellular, this sort of our this genome level things should not be manipulated. We should not be controlling. So it's like we are playing God kill again, right? We shouldn't do that here, right? So let's see. A major quality assessment of the human genome sequence was published in 2004. 92% of the sampling exceeded 99.99. So, after they have completed the human genome project, they did a quality assessment. Then they saw 92% of the experiment was almost 100% accurate. Sorry, the process was also accurate. Then, Human Genome Reference Consortium is a international collective of academic and research institutes which experts in genome mapping, still working on the analysis of the data obtained. So you have right now uh, a consortium that is called as what? The genome. So they are the people sort of the in charge of the all the information related to the human genome. Genome Reference Consortium. Highlight that. Genome Reference Consortium. Genome Reference Consortium. They are still working in the analysis of the data. Okay, because a large amount of data was obtained. The sequencing of the human genome holds benefits for many fields, from molecular medicine to human evolution. Improved diagnosis of various diseases, identification of mutations linked to different forms of cancer, design of medication, and more accurate prediction of the effects. Because can you understand? All these benefits arise because we know all the genes in our DNA together with the proteins that they are making. Yes, everything you know. So therefore, you can see by, I mean, before you administer a certain new drug to a human population, you can predict, okay, this is going to have this effect on this gene. It is going to have this effect on this gene because you know all the genes. Got it? Another proposed benefit is the commercial development of genomic research related to DNA based products. Okay, so DNA based, there are drugs. Okay, so many things are there that are sort of are you can design based on our genome because now you know everything to be known about our genome. Then other genome projects such as E. coli, yeast, mouse, Arabidopsis thaliana, and rice. Okay, what else are we there? Amazing. Arabidopsis thaliana, that's a type of tree, type of plant. It's a mustard cress. I think so. They have published. Got it. Then, is there a mouse genome? Yes. Rat genome is happened in somewhere like 2002. Then mouse genome, maybe mouse and rat. I think that's different. Okay. Then roundworm. I mean, E. coli. E. coli was one of the first things done. Yes, it happened very early in 1997. Okay, because it's a bacterium, no? A yeast. Yeast was even done before that, 1996. Okay, first bacterial genome, Haemophilus influenzae, influenza bacteria. causing pneumonia. Yeah, so those are the things that happened, right? Gradually with time. Because why the rat or the mouse is the most complicated? Because it's a mammal, it's a chordate mammal, so very complicated. Therefore, this opened the way to comprehensive description of molecular activities of human cells and the ways in which these activities are controlled. This is central to the continued development not only of molecular biology and genetics, but also the areas of biochemistry, cell biology, and physiology. The Genome project will have additional benefits that at present can only be guessed at. Okay, what are the additional benefits that we are thinking that will come in future, right? So that we think the benefits that we will be able to experience in future. Can you see? First thing, it is evident that the human genome, in common with genomes with many other organisms, contain extensive amounts of intergenic DNA. Very, very important. Now, by now, you know what is intergenic DNA. No? So, if you take a piece of our DNA, okay, genes have empty spaces. No, if this is gene 2, if this is gene 1, you know there's a segments of genes sorry, DNA in between genes. We don't know the function yet. These are called intergenic DNA. No? You don't know what the function is. Yes, so we still have to discover. It is thought that most of the intergenic has no function, except occasionally it is known that some of the intergenic DNA act to control genes nearby. So they can sometimes have a controlling effect on this gene or this gene. Okay, but we don't know it. Could the intergenic DNA have a role, but a role that is present too subtle for us to grasp? That means it could have a hidden function that we haven't yet discovered. Okay. The first step in addressing this probability is to obtain a complete description of the organization of the intergenic DNA at different genomes, so that the common features that might indicate a role for some of all these sequences may be identified. So what we have to do is we have to identify all the intergenic DNA in our genome, and then you can compare. Okay, this in this place there is intergenic DNA, so what could be the function? Okay, we have a still long way to go. Okay, so I'll send this PDF if you don't need to check it. Medine Francis, since the day Gregor Mendel discovered the laws of genetics, and then you have Fortson and Crick with the double helix model. Then any other people that you know from your syllabus? Recombinant DNA technology was invented. PCR was invented. Yeah, those things those things happen as the human genome project is going along. Yes, got it. Right. We'll do a few questions. Then we have only one final topic to discuss, that is stem cells. All right. Make Kanban Taggala. Question number 6. Okay, that's the answer. One of the aims of the Human Genome Project. Yeah, what was the answer? Okay. Determine the sequence of 20,000 base pairs. Do you have 20,000 base pairs? You have billion, eh? How many billions? Yeah, how many billions? How many billion do you have? 3 billion, no? Yeah, 3 billion base pairs are there. How many? What is the 20,000? 20,000 genes, protein coding genes. Yes. So that is 4. Human Genome Project is yet to be completed. No, it was completed in 2003. Yeah, almost 23 years have passed. Human Genome Project has led the description of molecule activities years. E. coli project is already completed. It is like a side project. Main mission game sort of a side mission was to complete the E. coli. Yes, because E. coli has less genes than us, much less complicated. So on the way, they completed mouse and all the other yeast and other genome projects as they are completing the human genome project. Why? Because they were developing tools. So using those new developing tools, they can easily sequence all the other genomes. 5 benefits of sequencing the human genome. So I told you, there's this paragraph. Ne? There's this paragraph in that page number 65. Can you extract 5 benefits? Can you extract 5 benefits that you can write? Why did we sequence the human genome? Why did we do that? 5 benefits that you got by sequencing the human genome. මොකටද මේක කරේ? Over almost how many years? 13 years. Why did you do this? Yeah. What are the benefits that you? Yeah. 5 benefits. Improved diagnosis of various diseases. Yes. Improved diagnosis of various diseases. Identification of mutations. To related to different forms of cancer. You can write. Then medication designing of medication, new drugs, and more accurate prediction of their effects. Gene therapy we'll talk about in stem cells. Okay. Again, control systems for drugs, gene therapy and control systems. Study of human evolution and anthropology. Anthropology is how humans moved, right, from one place to the other during history. How humans move from one civilization to the other. Anthropology. So those are the things you can say, Why did we do this? Because you can, if you know the human genome, you can completely understand how the human body functions to most of the states. I mean, we still don't know many things, okay, but this was like the starting point. Okay, clear? Anything else you wrote? Any other benefits that you wrote? There are some additional benefits at the end, right? Commercial development of genomics related research that you can write. Okay, commercial development of genomic related research such as DNA based products. Okay, so those can be vaccine production, those kind of things. Yes, that can happen. And what else? Then all these additional benefits, right? So that can only be guessed at. Those things don't write because they are not complete answers. No? Okay. So finally, we have only stem cells to talk about. Okay. Applications of stem cells. I want you to write as a short note. Okay. So we'll go through what is stem cells first. I'll erase all these. Okay. So Filaria then okay nene. You know everything about microfilariae and the adult worms and everything. Okay. So come to page number 62. Online people, stem cell therapy. There's a definition. Okay. Stem cells are undifferentiated cells which give rise to the cells of the same type. Okay. Undifferentiated cells which can give rise to the cells of same type. So everything starts with this pluripotent stem cells. Can you see in this diagram? Follow this diagram as you are reading the resource. Okay. Pluripotent stem cells are found where? Where do you think pluripotent stem cells are found? In this diagram that I have given. Can we take a 5-minute break? I'll be finishing in maybe another 15 minutes. Okay. Yeah, we can go ahead, right? I'll be finishing at 7:15. Okay. We don't need a break, right? Okay, so pluripotent stem cells are found in when you were a zygote, and then these early embryological stages you had pluripotent stem cells. Then they sort of differentiated into your germ cells, and then from your germ cells you got all the other tissue lines, right? So can you see your sort of immune system precursors, then your muscle cell precursors, liver, cardiac, nerve, intestinal, all those other precursor cells came from your pluripotent stem cells that you had in your embryological days. Okay, so that's what one of the main stem cells that we are going to talk about today. Okay. So, what is the definition? Undifferentiated cells which can give rise to cells of the same type. They can divide by mitosis without a limit, at least for the lifetime. Highlight that they can divide without by mitosis without a limit. Okay, they are capable of terminally differentiating. So you can see they have terminally differentiated into plasma cells, memory cells, or again nerve cells, platelets, red blood cells, cardiac cells. Right. So they are capable of differentiating. Stem cells are required wherever there is a recurring need to replace differentiated cells that cannot themselves divide. So, can you understand when a certain cell that is produced at the end, it is not able to divide by himself or to divide by mitosis, you have to generate him from a stem cell. So, your cardiac cells are produced initially, then they don't divide, no? So, they were produced from the early stem cells, and that was it. Okay, because later they are not going to divide by mitosis. So, for their formation, you needed a stem cell. So, the stem cell itself has to be able to divide, but it doesn't necessarily have to divide rapidly. Okay, in fact, stem cells usually divide at a relatively slow rate. Okay, so because stem cells are like they are making cells from scratch, so they are not. So, for example, I mean, if you take like a mitotically dividing cell, he has to only copy himself. No, he has to make a copy of himself. But the stem cell is a completely undifferentiated cell. So there you have to make a new cell from scratch, right? So you have to divide into a new cell, and then that cell has to undergo all these changes. So that's why stem cells divide at a relatively slow rate. Can you highlight that? Stem cells divide at a relatively slow rate. Stem cells are of two types, as embryonic and adult. Okay, can you see? So this is the embryonic stem cells. These are your embryonic stem cells. Okay, let's talk about embryonic stem cells. Following fertilization of a sperm and egg cell, the fertilized egg is a zygote. Zygote divides rapidly. In humans, around 5 to 7 days, the dividing cells create an embryo containing a small hollow cluster of approximately 100 cells called blastocyst. Blastocyst is approximately 1/7 of a millimeter in diameter. Okay, there is a small cluster of around 30 cells tucked inside the blastocyst that forms a structure that is known as inner cell mass. Now I think you can remember how in our study of human development we talked about this, right? We have this reproduction. I think I have. Uh, Reproduction tube, I had it somewhere. I don't think so. No. Right. I think in that tube, I can screen share, but I'll just draw. Right. A blastocyst is roughly produced after the implantation. I mean, before. So after implantation, the blastocyst is going to have two parts. That is going to be the inner cell mass. That is going to become the proper baby. So the inner cell mass has about 30 cells. That cells are what we call as embryonic stem cells. Those inner cell mass 30 around cells are your embryonic stem cells. The rest of the blastocyst is going to become what? The rest of the blastocyst is going to become what? The baby's part of placenta. This is going to become the baby's part of placenta, that is called as trophoblast. Yes. Trophoblast, that is going to later become the baby side of the placenta. So, this 30 cells of the inner cell mass is the embryonic stem cells. Can you highlight? Inner cell mass, around 30 cells, they are the source of embryonic stem cells. Can you highlight? The inner cell mass is the source of embryonic stem cells. Embryonic stem cells are so special, they can eventually differentiate into form all of the more than 200 cell types that make up the human body. Therefore, ES cells are called pluripotent. Can you highlight this in the tute? These are pluripotent stem cells, which are embryonic stem cells. Your pluripotent stem cells are your embryonic stem cells. Human embryonic stem cells are unspecialized cells with two major properties. They can self-renew indefinitely to produce more stem cells. Under proper growth conditions, they can differentiate into a variety of mature cells. So, can you understand? For some time, these pluripotent stem cells can produce more stem cells as well. They can produce more stem cells at the same time they are differentiating. So they are able to produce more stem cells, and the stem cells are going to differentiate into more and more different cell types. It could be blood cells, it could be nerve cells, muscle cells, liver cells, intestinal. All those cells arise from embryonic stem cells. Okay. Adult stem cells. Adult stem cells are much more simple and have less this quality that we call as pluripotency. This pluripotency is not found in adult stem cells. Let's read. They are found in the body, throughout the body after development. Highlight that. They are found throughout the body after development. Adult stem cells are present in many tissues. There are many types of adult stem cells specialized for genesis of different classes of terminal differentiated cells. Hence, each type of stem cell serves as renewal of one type of tissue. Yes, can you see? Particularly, adult stem cells are found relating to blood cell production. Okay, because you know your muscle cells, nerve cells, and cardiac cells are usually terminally differentiated. They don't divide again. Okay, so more than for those kind of cells, you have adult stem cells mainly for tissues like blood cell production. Okay. Epidermal stem cells for epidermis, intestinal stem cells for intestinal epithelium. These are then hemopoietic stem cells. So you can write a word here. These are called hemopoietic stem cells. The stem cells that produce blood cells. Write down. They are called hemopoietic stem cells. Poietic. Okay, poietic. Hemopoietic stem cells. They are the adult stem cells that are producing blood. Okay. Then neural stem cells for the neural stem cells for the nervous system. Okay. Stem cells can be isolated and cultured in culture media in vitro. You can culture stem cells in culture media because they are able to divide. Okay. When adult stem cells are removed from the body and maintained in culture or are transplanted from one site in the body to another, they generally remain faithful. Highlight that. This is what happens when you have bone marrow associated diseases. Okay. For example, a person who's having maybe uh, leukemia or something, they have bone marrow stem cells are there. I mean, they don't properly divide, so they are not going to have enough blood cells. So what you can do is you can transplant these stem cells into their bone marrow. So from a donor, you can take hemopoietic stem cells and put them into this deceased person's bone marrow, and they will produce new bone cells. Got it? That's why they remain faithful. Okay, so if you transplant hemopoietic stem cells into another person's bone marrow, they are not going to produce maybe epithelial cells, right? Intestinal cells. They are going to continue to produce blood cells. Okay, that's what they say. They remain faithful. In contrast, ES cells can be in contract. Okay, so that means like by law again. can be kept proliferating indefinitely in culture and yet retain unrestricted developmental potential. If stem cells, sorry, we are talking about embryonic stem cells here, ES. Okay. If ES cells are put back into a blastocyst, they become incorporated into the embryo, can give rise to all the tissues. So, can you understand? Adult stem cells can only give rise to their assigned cell at the end. But embryonic stem cells, they will differentiate into all the cells in the body. Yes. There are ethical concerns. Then with this embryonic stem cell, when you are experimenting or obtaining embryonic stem cells, there are some problems. Because why? What is the only source of obtaining embryonic stem cells? You have to get it from a very young, 7 day to that sort of age, small baby. Yes, that is the only way to obtain them. Therefore, research on human embryonic stem cells are very controversial. They are so because of their source, an early embryo. Scientists demonstrate successful derivation of pluripotent stem cells from adult tissues by reprogramming. So, one thing we can do is by you can reprogram adult stem cells to behave like embryonic. That is done by gene technology. Okay. These are known as induced pluripotent. So, you can have something called as induced pluripotent stem cells. You can have induced pluripotent stem cells. They hold promise in the field of regenerative medicine because they can propagate indefinitely as well as give rise to every other cell type in the body. So, what you are doing is you are going to sort of induce the qualities of embryonic stem cells in adult stem cells by reprogramming them, such as neurons, heart, pancreas, and liver. They represent a single source of cells that can be used to replace those lost to damage or disease. Since induced pluripotent stem cells can be derived directly from adult tissues, they not only bypass the need for embryos but can be made in a patient match manner, which means that each individual could have their own pluripotent stem cell line. So, what you can do is you can obtain stem cells from an adult, then you can reprogram it so that it behaves like embryonic. So what are the two problems that it eliminates? You can use it like your own spare parts. Can you see? You can using your own stem cells that you modify, you can use it as a spare parts for your own replacement. These are very highly experimental things. Okay. And the other thing is you don't have to kill a small embryo to obtain embryonic stem cells. Understood? So, applications of stem cells, I want you to write this as a short note. Okay, we'll check this next week. Short note. Okay, we'll check this next week. I'll give you a marking scheme. Applications of stem cells. Applications of stem cells, I want you to write. Okay. There are a few points. I will be checking this short note next week. Okay. So, from next week will be the revision class will be I am going to do a question based revision of some of the topics and I am going to give you some guest essays as well. I am going to be discussing some guest essays as well that can be questioned in the paper. Okay. So, join for those sessions until the exam is very close. Okay. So, let's see. We'll try to do the maximum amount of work until then. So, at this point you are collecting marks. Okay. You have the knowledge. What you have to do is so that you have to learn to gather that knowledge in order to go to the exam with a bag full of knowledge. Okay. So, your knowledge is everywhere in the room and in your brain, everywhere. So, you have to gather it now, collect it, organize it, so you can take it to the exam. Understood? Okay. We'll stop there. You write this short note as homework. Any questions? Can a person donate their stem cells? it's difficult to cross match because there's a high chance they can be rejected.